Hi, my name is Naomi Gobin.
And my name is Ariella Golbin.
The focus of our research is to identify the signaling mechanism by which a plant responds to a stress hormone.
Abscisic acid, or ABA, is a phytohormone that mediates responses to abiotic stresses such as salinity, drought, and low temperatures. Abscisic acid also plays a role in regulating stomatal closure, which controls the amount of water that leaves the plant through transpiration. Abscisic acid is, however, an unstable molecule, meaning it is hard to study. Due to this fact, a synthetic agonist, which means a substance that carries out a similar role to ABA, called pyrobactin, was discovered. The discovery of pyrobactin led to the identification of ABA receptors.
In the model seed plant Arabidopsis thaliana, there are 13 ABA receptors. When ABA is produced, it binds to the receptor and phosphorylates a kinase called SNRK2. This leads to the phosphorylation of transcription factors and turns on ABA-responsive genes such as late embryogenesis abundant proteins, which regulate water production.
Though this molecular mechanism is well understood in seed plants, it has not yet been characterized in seedless plants such as ferns. Seedless plants such as the resurrection fern have been proven to be tolerant to high levels of salinity and drought. Understanding the mechanism by which these seedless plants tolerate these abiotic stresses can aid in crop production. In order to study the ABA signaling pathway in seedless plants, we studied the fern Ceratopteris richardii.
Ceratopteris richardii is a fern that alternates between two free-living stages. The first is the haploid gametophyte stage, which is equivalent to the pollen and ovule in seed plants. This stage develops as a flat two-dimensional surface, which allows for easy access to cell division. The second stage is the diploid sporophyte stage, which is the typical three-dimensional stage seen in seed plants.
We focus on the gametophytes of Ceratopteris richardii because the pollen and ovule germination was severely reduced under high salinity and drought conditions.
Our research question is how pyrobactin is perceived in the development of Ceratopteris richardii. Our hypothesis was that pyrobactin will inhibit growth and development of Ceratopteris in a dose-dependent manner, and perception of pyrobactin will be mediated through conserved receptors such as PYL or RCAR.
In order to test our hypothesis, we imbibed the spores in the dark for 7 days and then induced germination by exposing the spores to white light for 3 days. After this, we exposed the spores to concentrations of 10 and 20 micromolars of pyrobactin and allowed them to develop under continuous white light. In order to test our physiological changes, we measured the germination of the spores of Ceratopteris, the area of the porthalis, and the number of cells in the gametophytes.
When treated with pyrobactin, we found that regarding germination, only the 20 micromolar data was significant. However, when looking at cell area and cell number, we found that both the 10 and 20 micromolar data was significant. This suggests that a higher dosage is needed to affect germination, while a lower dosage is able to affect cell division. In seed plants, pyrobactin inhibits the germination of seeds and hypocolla growth, which suggests that the ABA recognition mechanism is similar in seed and seedless plants.
The next part of our experiment was to determine how pyrobactin is perceived in Ceratopteris. To do this, we first identified the PYL receptors in Ceratopteris. We screened an express sequence tag cDNA library, which is a library containing mRNA sequences transcribed into cDNA. After this, we used basic local alignment search tool, or BLAST, to identify a single PYL receptor in Ceratopteris that had approximately 52% homology to Arabidopsis thaliana. We also carried out phylogenetic analysis, and we found that PYL receptors evolved before the separation of seeds from seedless plants. CRPYL was divergent from the PYL receptors in seedless plants. However, CRPYL is the most similar to the PYL7 receptor in Arabidopsis.
In senescence, as Arabidopsis ages, the expression of PYL7 increases. As we cannot study senescence, we focused on abiotic stress, specifically salinity. We treated 14 Dale gametophytes with 150 millimolar of NaCl for 3 days, isolated RNA from gametophytes, and did an RT-PCR. We found that similar to seed plants, salinity induces the expression of this ABA receptor in Ceratopteris.
In conclusion, pyrobactin inhibits the germination of spores of Ceratopteris richardii in a dose-dependent effect. In addition, CRPYL is upregulated by salt stress, proving it may have a similar function to orthologs of seed plants.
Our hypothesis was supported. For future studies, we would like to identify and characterize other paralogs of PYL receptors in Ceratopteris and investigate the effect of various stress conditions on the expression of these receptors. We would also like to identify other factors in the ABA signaling pathway and to identify and characterize mutants that are insensitive to ABA.